cell types that make up our bodies. Along the way, we will examine the pivotal role that stem cells play in the process. The story of the embryo is grounded in chemistry, as all the information needed to make an entire animal is stuffed into the DNA genome of the zygote. Our journey will thus explore how our understanding of genes grew from earliest concepts to the current state of our knowledge. We will scrutinize fundamental questions regarding genes and development. What happens to the genetic information that cells no longer need? How do cells overcome the tyranny of genes, enabling them to acquire specialized characteristics in the face of a uniform set of genetic instructions? How does epigenetic regulation, heritable changes that don't involve our DNA sequences, determine a cell's very identity? In the end, we will consider where this science of the embryo is leading in terms of our health. The emerging field of regenerative medicine, based on our nascent understanding of how tissues are molded in the embryo, has the potential to revolutionize how we treat a variety of diseases and conditions. We will consider the unsettling similarities between embryos and tumors, and how studies of development are leading to new cancer therapies. We will examine the causes of organ failure and contemplate why some organs and organisms are capable of regenerating while others are not. And we will eavesdrop on the promising efforts that are underway to apply lessons from the embryo to medical practice. These include ominous-sounding processes like cellular reprogramming and gene editing that have the power to create artificial tissues or rewrite our genomes. There is a danger in writing this sort of book. On the one hand, it risks venturing into territory that is overly technical. Each of the topics we cover, cellular differentiation, morphogenesis, the establishment of form, genetics, stem cell biology, could fill volumes, and it is hard to discuss molecular embryology without slipping into jargon. This recalls the warning that physicist Stephen Hawking received while writing A Brief History of Time that each equation he included in the book would cut sales in half. On the other hand, there is a risk of oversimplifying the science, or worse, indulging in exaggeration that results in some fantastical image of a utopian or dystopian future. Indeed, we will tread that line by focusing on the foundational discoveries that built and continue to advance our knowledge. The title, From One Cell, refers to two different but related types of cells with extraordinary potential. The zygote, which gives rise to every cell in the body during embryonic development, and the embryonic stem cell, which has a similar capacity. It is common knowledge that scientists hope to use stem cells to treat a variety of diseases, but what is less widely appreciated is that most of what we hope to achieve with these cells, or their surrogates, the embryo accomplishes as a matter of routine. Thus, for regenerative medicine to deliver on its promise, our best bet is to look inward, to see how our bodies and those of our phylogenetic predecessors were originally fashioned. In short, we need to understand normal development. After more than 2 decades of work in the field, as a physician, developmental biologist, and now cancer biologist, I am still awed by the beauty, mystery, and universality of the embryo. Yet as a practicing doctor, I also view the embryo through a clinical lens, given its potential to deliver remedies for myriad diseases. It is the nature of research that any advance raises dozens of new questions. Indeed, this self-perpetuating knowledge gap is what makes science so irresistible. The story of embryonic development, stem cells, and regeneration is therefore still being written. Even so, it is a compelling account involving scientists, philosophers, patients, and doctors. But the star of our narrative is the embryo, the entity from which all animals, including human beings, arise. From one